My paper is called The Evolution of Phenology in a Salmonoid Population, a Potential Adaptive Response to Climate Change. On this slide is an Ock Creek pink salmon, the group that this paper focuses on. Before I start, there are some important terms used throughout this paper. First, a fry is a juvenile fish. Second, phenology is the study of the timing of biological events. In this case, the major life event I will be referring to is salmon migration.
In the case of different salmon species, there is evidence of directional selection against the late migrating phenotype. In the auk species of salmon, the late migrating phenotype went from being found in more than 50% of the population to approximately only 10% of the population. This has persisted over the span of 13 generations, possibly reflecting adaptation to the warming of the water. More genetic data is needed to conclusively determine if this is truly adaptation and evolution. As climate change continues to become a huge environmental issue, it is important to see how animals are being impacted. This paper will address 4 questions. Before continuing, I want to point out what one of the abbreviations used often in this slide set is. LMMA stands for the late migrating marker allele. It has little influence on salmon life outcomes, but it has a high correlation with the late migrating phenotype. This makes it useful for genetic study. The first question we will be answering is, are these changes in phenology of Ock Creek pink salmon due to natural selection? The second one is, are these changes related to climate change? The third question is, have fry exhibited changes at the LMMA that are parallel to those observed in adults? And the fourth question is the low fitness of late migrating fish due to low marine survival or poor reproductive success? The hypothesis that this paper will be testing is the change in migration time of salmon due to the warming of the Alaskan waters. In this experiment, the methods that were used is salmon with late migration times were selectively bred and then marked by the existence of the LMMA. This allowed scientists to track late migrating fry. During each spring from 1984 to 1922, daily counts were made of all pink salmon fry that passed to the Ock Creek during their downstream migration. The genotyping was accomplished by using starch gel protein electrophoresis. If gene flow was occurring, the frequency of LMMA would decline within the late migrating subpopulation, but remain stable throughout the population. Conversely, reduced fitness of late migrating fish should have caused a reduction in the population-wide frequency of the LMMA. Now let's discuss the results of my paper. The LMMA frequency remained stable until after 1990 when it dropped significantly. These patterns indicated that the LMMA frequency was reduced by a selective event, following the fry immigration of 1990. Late migrating survival declined and only 1.4% of late migrants survived. And in 1993, 2001, and 2011, the salmon population was analyzed again, just to make sure the results were good, and the low frequency of LMMA persisted.
So in this graph, the fry are the open circles, which migrate on even years, and the adult are dark circles, which migrate on odd years. The x-axis is migration year, which you can look to see the odd or even year. And the y-axis is the late marker allele frequency. The higher you are on that axis, the more frequent that allele is. And you can see that for 4 generations, the LMMA was relatively consistent, relatively constant. You can also see based on the error bars, there was no really significant difference. But after 1990, there was a huge drop in LMMA, or a very significant drop in LMMA. This figure helps contextualize the previous figure. On the y-axis, there's fry abundance in thousands, so multiply the number you see on the y-axis by 1,000. And then on the x-axis, it's the calendar date. So later in the year, you can see that it is generally warmer.
The early migrating fry are the solid line, and the late migrating fry are the dashed line. You might be surprised to see that the biggest temperature difference is actually 1986, which you can see because the two lines are very separated from each other. Even though it was the biggest difference in temperature, it's not when the selective event occurred where early migrants were favored. This is actually because if it's too early, there won't be enough prey. The temperature will be too cool. So there's a balance between migrating early being beneficial if the temperature is still too cool. These results can be attributed to high predator vulnerability for late migrating fry. Late migrating fry tend to be smaller compared to the early migrating fry, because they haven't been able to eat as much prey, which makes them more vulnerable to predation. This paper ruled out gene flow to explain these results because they observed the late migrating salmon ability to mate with other populations in early migrating fry. Moreover, genetic drift was ruled out because the rate of change of LMMA based on the population size was too high to be considered genetic drift. Most likely, this change in genetic composition was due to natural selection. Let's talk about other papers that help contextualize the salmon paper. So first, the great tit experiment in Oxford is important to mention. In this study, it was found that there was a strong correlation between earlier egg laying time in the great tits and earlier and warmer spring temperatures. So similarly to the salmon, climate change was affecting the behavior of these great tits. This paper focused on phenotypic plasticity affecting the behavior of the great tits. So it was not their genetic composition that was changing, instead it was their behavior that they were able to change based off of differing temperatures. It is likely that the salmon exhibited a lot of their behaviors because of phenotypic plasticity as well as genotypic variance. I think the paper would benefit from citing some of these resources or perhaps following up to test phenotypic plasticity so it can be ruled out or at least shown to also have an effect with genotypic variance. Another experiment that is important to contextualize is zooplankton, which have been shown to bloom earlier, and this happens to be the salmon's prey. This shows the butterfly effect that climate can have on an ecosystem. So the zooplankton are affected, and then salmon are affected because of the zooplankton. Another experiment that is important to compare is that other salmon are also migrating early. It's not just this species of salmon, it's affecting a lot of salmon. All in all, climate change is affecting animals and their behavior significantly.